Good afternoon. My name is Anthony Fotenos, and I'm a clinical team leader in the Division of Imaging and Radiation Medicine in FDA's Office of New Drugs. In the last session, Donna Roscoe spoke about oncology therapy in which patient selection hinges on whether the output of a companion device is interpreted as positive or negative. What if therapeutic decision-making hinges instead on pre-therapy administration of a pharmaceutical drug or biologic? What if, compared to information sourced from in vitro samples removed from the patient, part of the administered active ingredient disintegrates and emits nuclear particles or photons directly from sources of biodistribution within the patient? In this case, we're talking about radiopharmaceuticals, the subject my colleague, Donika Plyku, and I will be discussing together over the next half hour. I will provide an overview of radiopharmaceutical regulation from a clinical perspective, and Donika will go more in depth on scientific aspects unique to radiopharmaceuticals and with a focus on early-phase clinical investigation. How does FDA regulate manufacturers' intent on new radiopharmaceutical marketing? The short answer is the same way as any other pharmaceutical drug or biologic manufacturer. For example, if you know that the Center for Device and Radiological Health, CDER, leads review of linear accelerator, brachytherapy, PET, and gamma camera devices, you might wonder whether CDER also leads radiopharmaceutical review. The answer is no. The Center for Drug Evaluation and Research, CDER, leads radiopharmaceutical review, though internal cross-center collaboration is common. This means that everything you have learned today from my colleagues in the Office of Oncologic Diseases about getting to and designing first-in-human trials for small molecules and biologics generally also applies to development of new oncologic therapy radiopharmaceuticals. Okay, so if in the main, everything you need to know about developing new drugs and biologics also applies to radiopharmaceuticals, are there any additional FDA applicable laws or regulations unique to radiopharmaceuticals? The answer is yes. A few regulations specifically focus on the tendency of radiopharmaceuticals to emit energy detectable at a distance. These regulations fall into two buckets. The first bucket involves dosimetry, requiring radiopharmaceutical manufacturers to investigate how much energy is absorbed by critical organs for safety evaluation. Dosimetry regulations apply both to therapeutic and diagnostic radiopharmaceuticals. Again, my colleague Donika Plyku will be talking about these next. The second bucket of radiopharmaceutical regulation applies only to diagnostic radiopharmaceuticals. Diagnostic radiopharmaceuticals are defined as those intended for in vivo administration, for the diagnosis or monitoring of a disease or disease manifestation. Internal to CDER's Office of New Drugs, review jurisdiction is organized by efficacy endpoints and therapeutic area. So the Office of Oncologic Diseases leads review for new oncologic therapy radiopharmaceuticals, and the Office of Specialty Medicine leads review of new diagnostic radiopharmaceuticals spanning multiple therapeutic areas. I'll next provide an overview of FDA's regulations specific to diagnostic radiopharmaceuticals because these are binding and likely to inform some of the product-specific advice FDA may provide via the formal meeting process for anyone interested in developing these products. United States Code of Federal Regulations, Title 21, covers food and drugs and is divided into Parts 1 through 1499. Part 315 covers diagnostic radiopharmaceuticals and immediately follows the parts that cover new drugs, investigational new drug applications, and applications for FDA approval to market a new drug. Under Part 315, the section that covers evaluation of effectiveness calls for focus on the ability of a diagnostic radiopharmaceutical to provide useful clinical information in comparison with reliable assessment of actual clinical status. Thus, in my opinion, it's never too early to ask one simple question when developing a new diagnostic radiopharmaceutical. What method should I use to find actual? In other words, independent of my product, what method should I use to find the clinically useful true information my radiopharmaceutical is also intended to provide? The Part 315 regulation essentially outlines 3 pathways for addressing this important question. First, if my radiopharmaceutical is indicated for localizing anatomical structures or characterizing their anatomy, I don't need to find an independent truthing method because all patients share a common anatomical architecture and human anatomy is well established. Thus, medical products that localize or delineate structures don't require truthing methods. Under this first pathway, by phase 3, I can design an adequate and well-controlled study in which imaging readers rate how well they visualize objective features of pre-specified structure. If there's agreement that such visualization is clinically useful, then demonstrating that reader ratings improve because of radiopharmaceutical administration will serve to satisfy the regulatory standard under this pathway. Under the second pathway, if my radiopharmaceutical is indicated for assessment of patient function or detection of disease, actual function or disease status is unknown. It must be established for each patient as a prerequisite for adequate and well-controlled investigation of radiopharmaceutical performance. As truthing methods, investigators most often rely on diagnosis provided by pathologists or on patient follow-up. Since all information can be represented as binary, I think of this pathway as the 2x2 table approach toward marketing of a new diagnostic radiopharmaceutical. Since this pathway is by far the most common, I'll provide 2 recent examples relevant to oncology. FDA recently approved gallium-68 Donika, a diagnostic radiopharmaceutical for PET imaging to detect somatostatin receptor-positive neuroendocrine tumors. This table is excerpted from the prescribing information. It shows the performance of central readers asked to interpret imaging as either positive or negative for the presence of neuroendocrine tumor. Gallium-68 Donika body imaging was acquired from 225 patients with known or suspected neuroendocrine tumor. The patients were referred to imaging for diagnosis or for evaluation of disease extension before or after treatment. Reader performance is quantified against the composite reference in the subset of patients with histopathology or up to 1 year of patient follow-up. All elements of the composite were pre-specified and prioritized according to the reliability of composite elements. Lower bound 95% confidence interval reader performance for co-primary endpoints of positive and negative % agreement was found to exceed clinically useful thresholds. FDA also recently approved gallium-68 PSMA-11, a diagnostic radiopharmaceutical for PET imaging to detect PSMA-positive prostate cancer. This table is again excerpted from the prescribing information. It shows the mean performance of central readers asked to interpret gallium-68 PSMA-11 imaging as either positive or negative for the presence of pelvic metastasis. Body imaging was acquired from 325 men with biopsy-proven prostate cancer referred prior to prostatectomy. Reader performance was quantified against histopathology in the subset of patients who proceeded to template pelvic lymph node dissection. Lower bound 95% confidence interval reader performance for co-primary endpoints of sensitivity and specificity was found to exceed clinically useful thresholds. Returning to the Part 315 regulations, for diagnostic radiopharmaceuticals to manage patients, usefulness may be demonstrated by improvement in how patients feel, function, or survive. The issue here is that FDA has yet to approve any diagnostic radiopharmaceutical for a patient management indication. This is likely because diagnostic products, by definition, are designed to provide information, meaning causal credit is assigned directly to the downstream product or procedure immediately proximal to the improved outcome, say, for example, administration of a therapeutic radiopharmaceutical. FDA has approved multiple radiopharmaceuticals for treating patients with cancer, including based on improvement in overall survival in patients with castration-resistant prostate cancer and based on improvement in progression-free survival in patients with neuroendocrine tumors. So here's where the concept of theranostics enter the picture. In the context of radiopharmaceuticals, a theranostic pair refers to therapeutic and parallel imaging radiopharmaceuticals that share a common mechanism of action. Given this tight coupling, oncology and radiation medicine review teams have been increasing internal collaboration aimed at minimizing waste of clinical evidence generated under investigations of new therapeutic radiopharmaceuticals when the same clinical evidence can be reused and leveraged to support parallel imaging radiopharmaceutical or peer approval. Based on this collaborative effort, we anticipate that new drug applications or supplementary new drug applications for a peer drug or biologic may be among future candidates for a first patient management indication under Part 315. But if not reader ratings or 2x2 table diagnostic performance endpoints, what peer efficacy endpoints might be considered to support such a novel approval? The answer starts with the Therapeutic Investigational New Drug Program. Let's assume it remains on track to provide substantial evidence of favorable benefit risk according to the same standards as any other pharmaceutical intended to treat similar oncology patients. What might FDA look for in its peer review to support, for example, a new patient selection indication? In general, we're looking to reuse endpoints for the purpose of peer performance quantification, endpoints already in place in whole or in part under oncology therapy protocols. For example, in reference to early phase therapeutic radiopharmaceutical dosimetry, for a peer endpoint, we might recommend investigation into the magnitude of agreement to peer dosimetry, since more agreement better supports proof of concept. In reference to later phase peer positive versus peer negative reads, we might recommend addition of central readers for investigation of inter-reader, and site central reader agreement, since more agreement better supports the generalizability of an effective therapeutic. In reference to agreed oncology outcomes in registration trials, we might recommend pre-specified subgroup analyses focused on contrasts specifically of parallel imaging interests, such as peer positive versus peer negative, or peer more versus less positive, since lack of such contrast would suggest excluded patients may benefit from an effective therapeutic. Finally, to maximize leverage at any phase, we might ask whether the oncology protocol incorporates pathology or other follow-up diagnostic procedures that might also be parallel repurposed as diagnostic truth standards for support of additional diagnostic radiopharmaceutical indications under the standalone Part 315 pathways described earlier. These additional pathways may be of interest as fallback considerations in case therapeutic development stalls or fails. Specific peer development phases include early phase range finding for optimization of peer-specific dosing, administration, image acquisition, and reader or dosimetrist instruction parameters. For efficacy, phase 3 statistical analysis plans to justify pre-specified success criteria for peer-specific endpoints, for example, those evaluating central reader agreement with respect to the to-be-marketed patient selection criteria. And finally, in the ultimate prescribing information work product, whether in the form of 2 or more well-cross-referenced oncology and imaging agent PIs, or one multi-targeted oncology PI, the aim is to provide instruction for all oncology co-providers, inclusive of imaging physicians. In summary, from my clinical perspective in the Division of Imaging and Radiation Medicine, I've here provided an overview of federal food and drug regulations specific to development of new radiopharmaceuticals. These include dosimetry regulations applicable to all radiopharmaceuticals and Part 315 regulations specific to diagnostic radiopharmaceuticals. These Part 315 regulations encompass at least 3 pathways for efficacy determination, with the third pathway focused on patient management and applicable to development of theranostic parallel imaging radiopharmaceuticals. This final slide provides references to additional reading on this subject for those interested. With that, I'll hand off the baton to my colleague, so my colleague can cover the remaining dosimetry ground, which is primarily associated with safety evaluation and grounded in the scientific discipline of medical physics. Donika, take it away. Thank you.
Thank you, Anthony. Good afternoon. My name is Donika Plyku, and I am a medical physicist at the Division of Imaging and Radiation Medicine, Center for Drug Evaluation and Research at the FDA. Today, I will talk about early phase development of therapeutic radiopharmaceuticals and theragnostics. My objective in this talk is to provide information to assist sponsors in the design of early phase studies for the development of therapeutic radiopharmaceuticals used in oncology. Evaluation of radiopharmaceuticals requires estimation of absorbed doses in tissue, and I aim to provide an overview of dosimetry regulations and methods for both diagnostic and therapeutic radiopharmaceuticals. With these objectives, this is the outline of my talk. I will first introduce the radiopharmaceuticals and their growing role in a personalized and precision medicine era. Next, I will focus on the design of early phase studies to support first-in-human trials with therapeutic radiopharmaceuticals, including regulations and FDA guidances. Then, I will review dosimetry methods for both diagnostic and therapeutic radiopharmaceuticals, and specifically the merged formalism for internal radiation dosimetry and dosimetry for RPT, including alpha emitters. A radiopharmaceutical is generally formed by a combination of a radionuclide with a biologically active pharmacophore or a targeting molecule that binds to specific cancer cells or a specific target in the body. Such a compound could be injected, infused, inhaled, or ingested and then make its way into the bloodstream. The development of radiopharmaceuticals builds on natural affinity, and examples include iodine 131, which has been used since the 1940s for the treatment of and management of thyroid disease, or radium-223 dichloride for the treatment of metastatic bone cancer. Diagnostic radiopharmaceuticals are administered at subpharmacological doses and are used to portray physiology, biochemistry, or pathology in the body without causing any physiological effect. Therapeutic radiopharmaceuticals deliver radiation directly and specifically to cancer cells. So the first component, the radionuclide or the radiation-emitting isotope, determines the imaging and therapeutic properties of the radiopharmaceutical depending on the predominant emission type in the decay process. The imaging attributes permit external detection of the radiation emitted within the body. The radionuclide can also confer desired localization properties, such as in the case of radioiodine, which physiologically accumulates in the thyroid gland and associated lesions when administered into the patient. The overall chemical structure of the pharmaceutical component, on the other hand, determines the biological properties of the radiopharmaceutical, acts as a carrier for the radionuclide component, and determines localization and biodistribution. Let's now look more closely at the radionuclide component. Radionuclides decay by different emissions. The linear energy transfer, or LET, can be used to differentiate between them. So radioisotopes may decay by beta particles, which are elementary particles that are sparsely ionizing, have a low LET, and therefore more tracks are required for cell kill. A radioisotope may also decay by alpha particles, which are heavy nuclei that are densely ionizing. The kinetic energies of alpha particles are 10 to 100-fold greater than beta particles. However, because of the much higher mass, alpha particles tend to deposit their energy over a very short distance and have a much higher LET compared to beta particles. So just a few tracks can cause cell kill, and the DNA damage is irreparable. The table shows examples of beta and alpha emitters. Photons can penetrate through tissue and allow external detection of emitted radiation, so photons are very important for imaging and diagnostic nuclear medicine. Imaging radionuclides can be divided in two groups: those single photon emitters that can be used in planar or SPECT imaging, and positron emitters that can be used in PET. Examples are shown in the tables. When selecting a radiopharmaceutical for a specific application, one has to consider the physical properties of the radionuclide, such as the half-life, and imaging modality specifications such as the use of collimators in SPECT or planar imaging, sensitivity, which is much higher in PET compared to SPECT, and quantification, which greatly impacts the accuracy of dosimetry calculations. So, for radioactive drugs, the Code of Federal Regulations, Title 21, Part 312, requires that IND submissions include sufficient data from animal or human studies to allow a reasonable calculation of radiation observed dose to the whole body and critical organs upon administration to a human subject. This has a direct implication on the design of phase 1 studies, which must include studies to obtain sufficient data for dosimetry calculations, and therefore IND submissions for new products include observed dose estimates for human organs that are often extrapolated from preclinical biodistribution data. So why do we need to perform radiation dosimetry? We need a measure to predict potential toxicity, efficacy, and risk associated with exposure to radiation. And for radiopharmaceuticals, we are talking about internal dosimetry, which is more challenging than the dosimetry performed in external beam radiation therapy. Here we are interested in calculating absorbed dose, which is the amount of energy absorbed per unit mass in target organ and tissue, and this is the most closely related quantity to the biological effect associated with exposure to radiation. With optimal dosing for RPT, one can achieve good tumor response within the constraint of normal organ toxicity. The FDA has prepared several guidance documents to make recommendations for radiopharmaceutical drug development, and one of these latest guidances provides recommendations on non-clinical studies and specifically proof of concept studies prior to a first-in-human study, preliminary characterization of the mechanism of action, and a pharmacology studies to determine the maximum tolerated dose in animals. For animal biodistribution and dosimetry studies, a single dose administration on a single animal species is recommended. All relevant information should be considered when selecting the animal species, and biodistribution studies are designed to evaluate radioactivity across time. Therefore, duration and sampling for data collection should be planned as needed. The purpose of such studies are to collect pharmacokinetic parameters to guide dose selection for the human biodistribution and dosimetry study. All daughter decays should be considered when designing the animal biodistribution study, and if any modeling is used, it should be described in the IND application. In addition, the studies should include aspects of the planned clinical biodistribution and dosimetry study to ensure consistency. For instance, if the planned clinical study includes patients being pretreated with thyroid-protecting agents to reduce radioiodine uptake by the thyroid, then the same design should be considered in the animal biodistribution study. Selected organs to assess biodistribution should include bone marrow and organs of excretion, and all organs specific to the radiopharmaceutical. In addition, both male and female organs should be included unless the indication is gender-specific. Excretion data should be collected, and for larger animals, these studies are done by imaging, so smaller number of animals may be sufficient. For smaller animals, this is done by animal sacrifice at different time points after injection. Organs are harvested and radioactivity is counted and used to characterize the biodistribution and time activity curves in animals. The measured percent injected dose per unit mass in animal tissue is then extrapolated to percent injected dose or activity in human organs, generally using a relative organ mass extrapolation that is shown here. This method assumes that the metabolism of radiopharmaceuticals is similar between animals and humans and varies only as a function of organ mass. So preclinical studies for RPT can project a rough normal organ dosimetry to help guide clinical trial design, but may underpredict the actual residence time and absorbed dose in human organs. These studies could be used to identify unexpected high uptake, and method of extrapolation from animal to human should be described in the R&D submission. Once we obtain the residence times in human organs, we can use the MERD equation to obtain absorbed dose in target organs. And to simplify this calculation, the MERD Committee of the Society of Nuclear Medicine came up with the so-called S values, which are pre-tabulated energy transfer coefficients that incorporate information on the properties of the radionuclide, organ anatomy, and organ mass. The S values are generated using Monte Carlo calculation based on standard human anatomy or human phantoms, and the picture on the top shows a very simplistic early model used for this calculation. However, more realistic and sophisticated models that vary in size and anatomy have been developed. So the absorbed dose to the target organ is the product of the residence time in the source organ with the S value from the source to the target organ in question, summed over all source organs. The recommendations in this guidance generally apply to new products with no previous clinical experience. However, when there is experience with the radionuclide or the ligand components of the radiopharmaceutical being developed, the non-clinical program can be abbreviated as needed, and the first-in-human dose can be based on clinical data as appropriate. Clinical dosimetry studies can be planned based on or using small patient cohorts and appropriate number and distribution of imaging time points. This is not fixed as it depends on the pharmacokinetics of the drug. So rapidly clearing activity will require more frequent and early sampling, and slowly clearing activity can be measured with fewer and widely dispersed samples. One can refer to relevant quantitative data for similar radiopharmaceuticals obtained in human and animal studies when planning a clinical dosimetry study. So to perform radiation dosimetry based on direct measurements in humans, the MERD methodology starts with the acquisition of serial quantitative images, which are co-registered across time. Then the next step is to define and segment regions of interest, and for each region of interest, one can characterize the time activity curve, which is model fit and integrated across time to obtain the residence time in source organs. This is then used as input in the MERD equation to obtain the absorbed dose in target organ, and this calculation can be done manually or using a software. So what I have described so far, this is the MERD formalism for internal radiation dosimetry, which was primarily developed for risk evaluation of diagnostic radiopharmaceuticals. It should be noted that this calculation relies on a reference geometry and anatomy that is a representative of a population and not specific to the patient. Therefore, tumor dosimetry cannot be directly handled within the MERD formalism. I described that this is an activity-based calculation based on phantom-derived S values. However, the phantom organ masses should be modified using available information from CT images when performing radiation dosimetry for radiopharmaceutical therapy. On the other hand, the voxel-wise techniques that pioneered in the context of external beam radiation therapy were adapted for radiopharmaceutical therapy and have evolved into commercially available dosimetry software. This is a dose rate-based calculation using Monte Carlo and patient-specific anatomy, where one can model voxel-level energy depositions for actual patient anatomy, including tumors. The table shows several software packages using these methodologies that have also obtained FDA clearance. The MERD formalism has also been adapted for alpha emitters. Here, the absorbed dose estimates depend on the pharmacokinetic fate and biodistribution of the daughters, so all daughters in the decay chain should be considered, and the difference in LET leads to a difference in biological outcome per unit absorbed dose. This difference is quantified by the RBE value, which depends on observed dose, biological endpoint, and tissue type. Dosimetry for alpha emitters should account for both organ level and microscale distributions, since alpha particles have very short range. So the organ level dosimetry can be performed using patient images. However, the microscale dosimetry requires a combination of both whole organ measurements and preclinical measurements in subregions of critical organs. These measurements can be implemented in the context of the MERT methodology, and an RBE-weighted dose value can be calculated using all emission types with corresponding RBE values. An assessment of late radiation toxicities is warranted when patients have a long life expectancy that could be affected by late radiation adverse events. The assessment could be based on animal and human biodistribution studies and publications on late radiation effects. Another FDA guidance document shown here provides recommendations to industry for designing non-clinical late radiation toxicity studies to determine the potential for late radiation effects. All radiation risks should be communicated in product labeling. So, the dose or administered activity for first-in-human studies should be selected based on tolerated radiation doses in human organs. However, currently, there are no accepted criteria for determination of organ tolerance for internal radiation from radiopharmaceuticals. Because of this, the sponsor should use published literature on external radiation therapy as a starting point for radiopharmaceuticals. In general, organs with actively dividing and less differentiated cells tend to be more radiosensitive, while those with low cell division and more differentiated cells tend to be more radioresistant. This list shows organs in decreasing order for radiosensitivity. In addition, the clinical significance of the organ is taken into account when deciding the maximum tolerated dosing or activity for clinical studies. To summarize, non-clinical dosimetry studies extrapolated to humans can be used as a first approximation for radiation absorbed doses in human organs to select a starting human dose. Human dosimetry studies provide a clinically relevant estimate, and dosimetry for RPT has its roots in the formalism established by the MERT Committee of Society of Nuclear Medicine. The federal regulations outline requirements for non-clinical and clinical dosimetry for early phase studies. Additional considerations, including a more patient-specific or personalized dosimetry approach for radiopharmaceutical therapy, including voxelized calculations and human tumor dosimetry, have been noted. And early and frequent interaction with the FDA is recommended when designing early phase studies with therapeutic radiopharmaceuticals. Thank you for your attention, and Anthony and I are looking forward to addressing your questions.
Thanks, Anthony and Donika. You guys are probably pretty happy that this stuff is so far above my head I can't even ask a semi-cogent question, but someone else has asked a question that I wanted to ask, so hopefully it's not completely naive, and I'm gonna add a little flavor to it. I'm a small company out there, or academic investigators, and I managed to find an epitope on mesothelioma, and it seems to be highly expressed on mesothelioma and not expressed on any other tissue. And I'm trying to figure out, do I want to make an antibody drug conjugate? Do I want to put a radionuclide on that? Do I want to turn it into a CAR T cell? And I mean, I would turn it into a CAR T cell. But I want to get a sense of, I understand when an antibody drug conjugate oncologists give antibody with a radionuclide on, I have to refer my patients to a radiation oncologist. There's some issues there, but I want to know what has the most punch, the most impact on that tumor. And if you guys had to pick a radionuclide, of all the ones that are out there, and I know that you guys have seen a lot. I've seen some very interesting. I've seen lead and copper. And what radionuclide would you pick if you were really looking for the greatest anti-cancer effect for your antibody, for your nucleide your radionuclide conjugated antibody?
Hi, Jeff. So I think what you're asking is what is the ideal radioisotope for oncology therapy, and that seems like a great question actually, because the distinguishing aspect of radiopharmaceutical therapy relative to the other cancer therapeutics is the ability to select the radionuclide, and in particular the type of radiation emissions that are delivered. And so when selecting the radionuclide, one has to consider certain physical characteristics of that, and that is, if I were to list them, that would be the physical half-life, which is known, and the type of emissions, which are also known for radionuclides. We know the decay chain, and then we can characterize and take them into account. And also the energy of the radiation and daughter products in the decay chain. Other considerations on this list would be the method of production and the radionuclide purity. So these are, I think, like the list of considerations for physical characteristics of radionuclides. Apart from this, one has to look at how readily available are these radionuclides. And so before I go there, let me explain a little bit these physical characteristics. So I would say a suitable range for therapeutic radionuclides for the physical half-life would be in a few hours to say 7 days or so. And because a very short physical half-life would limit the delivery flexibility and would make treatment impractical. A long half-life will retain the radiation dose in the patients and will expose surrounding surrounding people for long periods. So when a physical half-life is too long, patients need to be admitted and isolated for longer periods, which increases treatment costs. So I would say, but in addition to this, the physical half-life should be chosen in accordance with in combination with the biological half-life, because ultimately what we are using is the radiopharmaceutical. So what is important is the effective half-life, which is a combination of both physical and biological half-life. And this consideration is chosen to have an optimal effective half-life, which, when we are talking about the biological half-life, we need to consider the method of administration and then the uptake mechanism. So basically for radiopharmaceutical, we need to look at the kinetic compatibility between the radionuclide and the targeting molecule. So for small molecules have faster kinetics, but antibodies have slower kinetics. So if the physical half-life is long and the uptake is slow, then the radionuclide will not decay completely before it reaches the targeted tumor, right? So that will be, the first consideration, the half-life. Then one has to look at what I mentioned in my slide, the range of emissions. and the ionization density. So, and for that, I mentioned LET. LET is really the ionization density. And as I compared alphas versus beta particles, so the alpha emissions have high LET it is 400 to 500 times more than compared to beta particles. So this is the energy per unit length that is deposited in tissue. And in addition, the range of alpha particles in tissue is about 50 to 100 micrometer, so comparable to the cell dimension. And one is to, when looking at this, we need to think about the dose distribution. In beta particles will be smoothed because they have much longer ranges in the organ compared to alpha particles. And alpha particles, the dose distribution will be not, or is expected to be a non-uniform dose distribution. So, and then, when you consider LET, so ionization or the energy, deposited energy per unit length of this emitted radiation, then this translates into the radiobiological effectiveness of the radionuclide. And so alpha particles have higher RBE, have higher probability to cause cell kill, and because the energy or the emission can transverse the cell nucleus, so the DNA causes DNA damage, and because of this, an alpha particle is administered at lower administered activities compared to beta particles, and the factor between the two is about 1,000. So if a beta particle is administered, or what I'm saying is, if a radioisotope that emits beta radiation is administered at gigabecquerel administered activity, an alpha emitter is administered at megabecquerel administered activity. And the other thing is, when comparing these two, so the alpha particle would be, or alpha emitters would be better at for treating chemo and radioresistant tumor cells, and this is related to the radiobiology again. And it would be better or more ideal for single and small tumor clusters. So, these are some physical characteristics. And then, the other thing that I mentioned in my presentation is the daughter products. And all the daughter products in the decay chain should be considered, especially for alpha emitters, one has to take them into account when calculating absorbed dose. And then, in addition to this, I mentioned that beta emitters are more readily available, and then it depends on the method of production. So then I'll let Anthony add if anything I missed.
As Anthony said, scintillating. But I just I'm going to pin you down here. You have to pick a radionuclide. If you're a company, and there's no proprietariness here because it's, there's no patents on radionuclides, which one would you pick if it was your company?
So again, yes. So again, it's not, I would say, again, it would be like a consideration of characteristics. It's give and take a little bit, it needs to be balanced in the consideration needs to be balanced in the decision. But alpha emitters have shown efficacy in patients that were beta emitters when the cancer was recurrent after patients received beta emitter or treatment with beta emitters.
Thanks. Anthony, Dr. Fotenos, do you want to chime in there? We don't have much time, so this will be the last, the only question, and the last question. Anthony, if you're talking, we can't hear you. Type into the box.
Anthony, Anthony, if you can check to see perhaps your microphone is double muted. Otherwise, because it does show that you're unmuted in Adobe Connect, but we're not hearing anything. And it's looking like we may just need to move on because the.
Yeah. Thank you guys so much. That's the end of our time for radionuclides, and for me, it was almost special relativity as well. I want to thank you guys for participating, and more importantly, for all the collaborative efforts with your division. When the oncology division see these radionuclide pre-IDs and INDs, you guys are always there, and you're immensely helpful to help us understand this stuff.